Okay, this is a voice recording for enzymes. This will help connect to our textbook chapter 8, but generalized information about enzymes, how they function, how they're made, and the big picture of enzymes. Now, enzymes are catalysts. They are biological catalysts, and all that you really need to know about catalysts is that they speed up reactions. We'll explain how they speed up reactions in a little bit. For those of you that don't, that's shorthand for reactions. Okay, so they're going to speed up reactions. They're not a reactant. They're not a product. They don't get used up. They just speed up reactions. And we'll talk about how that happens. It's mostly with activation energy. So what is an enzyme? Enzyme is a protein that is made by a living organism. So that means that we're connecting back to ribosomes, and I'll talk more about this in the next slide. And the protein formation includes DNA information, so the transcription and the translation component as well. But here we have a generalized picture of an enzyme. So make sure you understand this for sure, and then how the labeling goes. So A in this picture is going into B, as you can see, and this B doesn't change through all three pictures. So that means that B down here is the enzyme. This enzyme is not going to change in our generalized view. A is the thing going in, and this is a substrate. And that substrate in this example here is getting broken down, so we're talking about more of a catabolic reaction. Enzymes can be anabolic as well in that they build up, so we can kind of reverse this with other enzymes. So here we see that this substrate is fitting in. Now it's very important that you understand that not only is this little groove here where the substrate fits in called an active site, but that enzymes are incredibly specific. That means that they work on usually one substrate. So every enzyme generally works on one substrate. Now I say generally because there is some flexibility based on the form of the substrate that's going in and the actual form of the active site. There's some looseness that goes in with that piece. Now, it's very important that you understand that enzymes generally, once again generally, are named after the substrate or the thing that they're working on. They have this ending that tends to be ASE. Doesn't always, but tends to be ASE. So we'll talk about some examples here and try to put it in perspective for you. But this substrate should fit in very, very, what we call specifically. There's this induced fit that when it comes in, there's this little microenvironment that changes, and maybe that enzyme flexes in on that substrate and causes the reaction to occur, generally loosening those bonds in between whatever covalent bonds that might be formed in that organic substance that we're talking about. So then it does its little work. You can see that the enzyme is available for another reaction if another substrate were to come in, and in turn you get these two things coming out. We call those things coming out the products. Now, big picture, how is this all happening? Well, we need to know how these enzymes are made. So enzymes, like I had said earlier, are proteins, which means that they're made out of monomers called amino acids. Now, amino acids are consumed by the body and used frequently. Proteins are much bigger polymers. So the ribosome, right, every single cell should have ribosomes, and there's a little asterisk in that statement that not every single cell has them, but generalize every single cell, bacteria, human cells, giraffe cells, plant cells have ribosomes. They're generally in pictures little teeny dots, but they actually have two subunits made out of rRNA and protein. So you got this small subunit and a large subunit. We'll have mRNA, right? If you remember anything about mRNA, it comes from the nucleus. So this is kind of like a copy of the DNA code. So if your nucleus is up here and you have your DNA code, right, double helix, that code gets transcribed into mRNA. There's a little bit of processing where we cut out some different pieces, modify that transcript. That transcript then comes out to the ribosome. The ribosome is the site where this protein assembly of amino acids is going to occur. We have some molecules that help bring in these amino acids called tRNAs, all things that we'll talk about later. But these tRNAs can bring these amino acids into this site, and it matches up with the code that is on this mRNA. It's usually read in triplet code, which we call a codon, and there'll be an anticodon on, I'm just going to generalize this shape again. That's my favorite word apparently today, generalize. There's this anticodon that matches up as a complement to the codon on the mRNA. And those amino acids in humans, in life in general, there are 20 different ones that all of life, for what we know right now, are available to build things. Now, proteins come from code then. That means in your code you have the very specific sequence that determines what that enzyme's amino acid chain is going to be. So those proteins then are very specific. Slight alteration in one little nucleotide up in here in that DNA will dramatically change sometimes the protein. So if it changes, if you refer back to that last image, if it changes that active site, we change one little amino acid, maybe this part of the active site pops up. Now our substrate can't fit in there. So things are very specific. An alteration of just an amino acid, of just a nucleotide, can alter how that enzyme looks, and its 3D structure determines its function. Okay, so make sure you really understand that piece, that form equals function. This is a common theme, something that you really need to understand, not only talking about isomers, but also talking about the human body, right? Bodies of other organisms, whether or not be human or not, all form equaling its function. All right, so what is this? This is sucrase, stolen clearly or borrowed from Shutterstock. So you can see that this is a very complex, colored, nice little structure. You can see, I don't know if you can see that well enough, but that's a little green twisty thing. That's an alpha helix, an amino acid chain that forms a secondary structure with some hydrogen bonds. You can actually see some of these what are called R groups sticking out here. Some of them stick out because they're polar. Some of them have to be kind of wedged in because they're non-polar, but they all form very specific shapes, and you can see that shape here of sucrase. So sucrase will work on the sugar sucrose. Right, now what is sucrose? Sucrose, basic drawing here, we have 6-carbon sugar that's holding hands with another 6-carbon sugar, because sucrose is a disaccharide. That glycosidic linkage there holds glucose and galactose together. And what happens then is if we end up getting a break in that, we end up seeing glucose floating away, and glucose can then be used by the body. I'm sorry, I misspoke, glucose and fructose. And fructose will be able to float away and be used by the body, and isomerase can change it into glucose if we need to change it. But it's very specific. Sucrase is really only going to work on sucrose. Sucrase will not work on another disaccharide called lactose, which is glucose and galactose. It won't break the bond, the glycosidic linkage there, because its active site is different. So even though sucrose and lactose have the same exact molecular formula, C24H22O11, they can't be broken by the same enzyme because they're slightly different. They're isomers of each other. You might be asking yourself, well, why isn't it the C doubled? Why didn't the H double and the O double like we would think? Well, remember that when we form disaccharides, we have to do dehydration reaction, pulling water out. If you notice, H2O is the removal of doubling from our C6H12O6. So water was removed. Sucrase, this enzyme right here, will allow for the addition, right, called your condensation reaction, where we bring in water, right, to actually break them apart, or hydrolysis to break them apart. Sorry, condensation would be pulling them out as well. But we're going to hydrolysize this by adding some water to break that apart. All right, so let's talk a little bit more about energy graphs. If we have energy on the y-axis here, we have to understand how enzymes pertain to this. We have time or reaction quotient on the x-axis. You can see two different types of graphs. One would look like this. Another one might look like this. There are commonalities in here, but slight differences. Always start with reactants on the left-hand side and products on the right. You always see this little hump here because that represents activation energy. And you end up having the products at a different level of energy than the reactants. Now in this one, the reactants have more energy than the products. So that means that we've lost energy, right? So we'd call this an exothermic or an exergonic reaction. Whereas this one, energy is being added, so we call that an endergonic or an endothermic reaction. Regardless, we're getting a change. Now, how do enzymes play a part? Well, activation energy is the energy required to get this thing to the actual products. If we add an enzyme, regardless if it's the graph here on the left or a graph on the right, it will decrease the activation energy. So we could do it more at an ambient temperature, a body temperature, if we're talking about enzymes in our body. Whereas if we didn't have the enzymes of sucrase, it would be very hard for us to react sucrose and turn it into the two products of glucose and fructose. It'd be much harder. An example of an endergonic reaction that we tend to see in biology would be the reactants of CO2 and H2O. This is not stoichiometrically balanced here, I'm just writing it, turning into C6H12O6 and oxygen. That's photosynthesis. The reverse, if we flip this around and throw it as an exo, that'd be cellular respiration. So make sure you understand how energy graphs work, how the enzyme plays a part, what activation is, what the reactants are, what the products are, and be able to classify exergonic versus endergonic. All right, last couple things here. What factors affect enzymes' rate? Well, concentration is a big part. Now we can talk concentration of enzymes, and we can talk concentration, enzymes, Y there. Enzymes, and we can also talk about concentration of substrate, and this relates to your lab. If we increase the concentration of either one of these, and we're talking about reaction rates here, increasing the concentration of the enzymes will increase the reaction rate. But eventually it gets to the point where it can't speed it up anymore. No matter how many enzymes you add, it gets to basically a velocity that's a terminal velocity, a velocity max that it can't go beyond. And substrate does the same thing. If we add more and more substrate, it'll work so fast and eventually it can't work any faster. The analogy that we've talked about is if you're building a house, enzymes would be your workers, your construction workers building the house, and the substrate might be the wood that's the house turns into. So the enzymes, if we have one worker at the site and 10,000 pieces of wood, it's going to take a long time down here for the house to be built. The rate is low. Right, so this would be rate over here on the y-axis. But if we add more workers, so enzyme concentration increasing, we should be able to increase the speed at which we can build that house. Now there's a point where you can't add any more workers to the work site to speed up the work. You're going to be limited. Same thing happens with substrate. If you get one piece of wood per day delivered to the site, it's going to take a long time, or the rate's going to be very low. Whereas if you add more and more pieces of wood, more workers can grab wood until the point where they can't grab wood any faster than them reaching out and grabbing that wood and nailing it together. So they're limited by the speed of that enzyme, and that's what you're seeing there where it plateaus out. Temperature, pH, and salinity have very similar types of graphs, and then if we have rate again on the y-axis, and we have either temperature, pH, or salinity down here on the x-axis, you generally see a bell curve for these, meaning that they have optimal conditions. Every single enzyme, remember they're very specific, every enzyme is going to be slightly different, where they have an optimal condition where they work the best, meaning that environment is very specific for that enzyme. If we talked about an enzyme like pepsin, which is in your stomach, it loves a pH of 1 to 2. But if pepsin, which it does, drops into your small intestine, where it tends to have a pH of 8, well, here is our optimal pH of 1 or 2. 8 is going to be way upstream here. You notice that the activity pretty much drops off as it moves upstream. Temperature is the same way. Your enzymes in your body love that 98.6 core temperature. You start moving it away, hypothermic or hyperthermic, and we end up running into issues. Salinity, salt concentration, same thing. Right? We have a very specific salinity in our body. We mess that up, it's going to cause big differences. All right, so make sure you understand those. Last two things is inhibition. Right? There's two different types of inhibition. If this is our enzyme, we're going to make this a person. So this is Eddie the enzyme, right? If this is our active site, and Eddie enzyme's job is to eat this hamburger. Put a little bit of lettuce on it. Sorry, no colors, folks. I'm going to try to keep this simple and fast. This hamburger fits right into Eddie enzyme's face. Competitive inhibition would be as if you threw a tennis ball that has the same type of shape into his mouth. That would inhibit or slow down the ability of Eddie to be able to eat this hamburger. Competitive, right into the active site changes. Non-competitive, it would be if we're talking about Eddie again. And here's his active site, right? We have the hamburgers that he's still working on as a substrate. Non-competitive is if you whip the ball at the back of Eddie's head and it causes his mouth shape to change. Now that hamburger is going to have a harder time and it's not going to fit in as well. So non-competitive, usually we affect a different spot other than the active site that slows down the speed at which that enzyme can work. We will talk about this in a second here, but that spot generally is called an allosteric site. All right, let's talk allosteric site and the negative feedback system. So allosteric site, like I just had mentioned, is this alternative spot on a different part. Well, I don't know what happened there to his face, but there you go. Now he's got a lip. Spot right here, which will sometimes the allosteric site can speed up how fast the enzyme works, helping induce the shape of the active site into a better conformational shape to work on the substrate. Or like I just said, it'll deactivate it. Now the deactivation part is really important for you to understand negative feedback. So if we have a chain reaction of A turning into B, turning into C, we have enzyme here on this line, X, and we have an enzyme here on this line, Y, negative feedback would be where this C product here, this final thing that we really want to make, will come back up here and when its concentration increases and allosterically impede, right here, if this is, well, we're changing Eddie's name to X, where C binds in here and turns off the enzyme. And the whole purpose is very similar to how the thermostat in your house works. If you want the thermostat in your house to read 70 degrees Fahrenheit, right, you don't want the thermostat to turn on the heater in your house, your furnace, and cause the temperature to continually rise forever, right? If your temperature in your house falls down to 68, the thermostat sends a signal to your furnace, causes the heat to go up until it reaches 70, and then it turns off. That's how things work in your body. You want insulin in your body. Your body doesn't continually make insulin. It makes insulin until the concentration is at the homeostatic level it needs to be at. If it goes beyond that, we run into issues. If it is under that, we run into issues. Homeostasis is the key. So negative feedback is the key way your body can keep homeostatic numbers in check. And that's super important for organisms like ourselves as humans. If you have other questions about enzymes, please feel free to email me. Otherwise, check it out in your book. I'll see you in class. Good luck.